My name is Sarah Swisher. I am an assistant professor in the Electrical and Computer Engineering Department here at the University of Minnesota. My own work is in flexible electronics for neural interfaces and other biomedical sensing devices. So I'm really excited to kick this session off this morning. We have some really great lineup. So there's a lot of interest and a lot of efforts both in scientific and commercial domains right now in leveraging technologies to interface with the brain and with the body for restoring function and treating various diseases. And these technologies are interfacing not just with the brain anymore, but with the body and other end organs, right? So I'm really excited to hear from three speakers in this area today. The first is going to be Dr. Florian Solzbacher. We'll then hear from Dr. Todd Coleman, and finally Hubert Lamb from here in Minnesota. Okay, our next speaker is Dr. Todd Coleman from the Department of Bioengineering at Stanford University. Dr. Coleman received his BS degree in electrical engineering as well as computer engineering from University of Michigan, his PhD from MIT in electrical engineering, and did postdoctoral studies at MIT in neuroscience. And the university, he's held faculty positions at University of Illinois and also UC San Diego. He's currently the U Family Faculty Scholar in the School of Engineering and an associate professor in the Department of Bioengineering at Stanford. His research is very multidisciplinary, using tools from applied probability, physiology, and bioelectronics. And he is selected as a National Academy of Engineering Gilbreth Lecturer, Fellow of the IEEE, Fellow of the American Institute of Medical and Biological Engineering. Currently chair of the National Academy's Standing Committee on Biotechnology Capabilities and National Security Needs. Dr. Coleman, thank you so much. We're very much looking forward to your talk.
Thank you. Hope everyone can hear me. And I just wanted to start off by saying that I'm a little bit of an outsider in this field, so I'll tell you a little bit about my story. I'm originally a quant by training, so my PhD was in electrical engineering with absolutely no interest in anything biological, developing applied probability algorithms for wireless communications. And my PhD advisor gave me the best piece of advice in my career when she strongly encouraged me to do a postdoc in something wildly different. And so I ended up doing a postdoc in neuroscience under Emory Brown. So I began to learn about physiology and neuroscience through the language of probability statistics. And then in my career, I also got involved in some technology development. And so our research endeavors sort of operate at that intersection of, we still actually do bread and butter analytics, my first love. Great opportunities now with these very massive data sets, thinking about how to do these types of, address these questions. We've also been involved in developing some flexible, stretchable electronics. Started off with my time as an assistant professor at Illinois when I was colleagues with John Rogers, and we worked on some things together and connecting everything to neuroscience and blending with analytics. Interesting applications within the context of sleep monitoring and whatnot. And so when I did that postdoc in neuroscience, there's a phrase that I call feeling comfortable, feeling uncomfortable. That's what that process was. And this has sort of appeared again because here's a picture of me and my dad at a family reunion, and I lost him to pancreatic cancer in 2011. And it turns out he had previously lost his mom to stomach cancer. So I knew there was a little bit of history of disease of the digestive system in my family. And so I told myself, I know absolutely nothing about the digestive system, but again, feeling comfortable, feeling uncomfortable, I'm gonna try to spend some of my time in this area. So when I first started to look into this area, I got a little worried because when I think about the digestive system, I think about chemistry and acids breaking down food. I'm not a chemist. I think about the mechanics of propelling food through the GI tract. I'm not a mechanical engineer. But then I discovered that we have 100 million neurons in our digestive system, and that began to excite me. And then I began to learn that the gut and the brain are connected not only via things like the HPA axis, but also through nerves electrically, like the spinal cord and the vagus nerve. So that was sort of my foray into this area, sort of completely by accident. So if we focus for a minute on the stomach, and then by the way, it's the most vaguely innervated digestive organ. The top part of your stomach has the fundus that sort of can expand a lot when food comes in. And then downstream, we have some pacemaker cells that I call the neuromuscular maestros because they integrate information from the nervous system of the gut, the enteric nervous system, and from the innervation from the brain. And they have ion channels, and so they can generate action potentials just like neurons. They go above threshold, so things that can increase the membrane potential include stretch, as I alluded to before, that elicits downstream neurotransmission, acetylcholine coming from parasympathetic innervation, and things that can inhibit or reduce the membrane potential are sympathetic innervations like norepinephrine and nitrous oxide. One unique thing about these cells is that their membrane potential is always oscillating. Even in the absence of any type of neuromodulation, it's always there. So the effect of that is that when these action potentials get generated, they're actually connected via electrical synapses, gap junctions, to the smooth muscle cells, giving rise to contractions. So what's kind of unique about this is if you look at not just a cell at one location, but if you look at them spatially, there's actually a traveling wave that's orchestrating this oscillating membrane potentials, and this basically orchestrates the timing of when the pinching is occurring, eliciting the propelling of food through the GI tract. So at a very high level, we got interested in it is just like how EKG can measure electrical activity of the heart, can we place electrodes on the abdomen to measure electrical activity of the digestive system? And so some people got excited about doing this in the 1980s, and originally they suggested basically just using 3 electrodes, which will generate one waveform. The other 2 electrodes are things like a ground and a reference. Now those membrane potentials that I alluded to of the pacemaker cells, they oscillate at a known frequency. For instance, in the human stomach, it's 0.05 hertz, so very low frequency. So at a very high level, what people came up with was, well, if I build a time frequency plot of what occurs, and let's take a look at when you eat a meal. So when you eat a meal, that's almost like a form of stimulation because the top part of the stomach is going to expand. It's going to sense that, send the downstream neurotransmitter. Do we see larger oscillations occurring? And that means do I see more red energy at around 0.05 hertz? So these types of ideas were explored in the 1980s, but then a lot of publications came out that basically said doubtful clinical usefulness in comparison to clinical gold standards. So people tried to do this but didn't get meaningful information. Fun fact about myself is I'm from Dallas, Texas, the spirit of the Dallas Cowboys or the Dallas Mavericks. Just because other people tried and failed, we said we're going to be some Cowboys and try anyway. So building upon, going beyond what was done in the 1980s, we discovered a couple of key things. One was that if you look at where they suggested placing the electrodes in the 1980s, we started to collaborate with gastroenterologists, and if you look from CT scans, the placement of the stomach relative to the exterior landmarks, there can be quite a bit of variability. So as you can see in the middle 2 plots, the 3 red dots are the suggestions from the 1980s, but you can see the stomach is much further above. So you can solve that problem with a multi-electrode array. Another issue is that there's a lot of motion artifact. When you place electrodes on the abdomen, you have a lot of musculature in the abdomen, but we can develop signal processing algorithms to deal with that. Now, something else that we got interested in are the fact that I mentioned that there's this spatial slow wave propagation, coordinating the timing, the spatial temporal aspects of contractions. If I just have a single waveform, I can't get spatial information. I need to look at multiple waveforms and look at the phase offsets. So we took advantage of wanting to use a multi-electrode array to not make sure we're covering the stomach, but also to capture the spatial propagation information. And so as you can see with these 3 different colored waveforms, those are waveforms from electrodes at different locations, and it turns out we can visualize the slow wave non-invasively, and we can do a variety of signal processing to look at the direction the wave is going, to make sure, remember I'm originally a quant, so we have to make sure that what we're picking up is not a consequence of just noise, so we can develop statistical procedures to do that. And in doing so, we can basically look at things like, so what's the spatial histogram of statistically significant wave propagation, and is it doing what we expect, which is always propagating, down the GI tract, so towards the small intestines. So we were able to get IRB approval and do some studies in human subjects, and a couple of interesting things occurred. If we take a look at patients where we have imaging, in a patient-specific manner, we can look at the direction towards the small intestines. And so, for instance, in the left column, we have a human control, and towards the small intestines is about 175 degrees or so. And when we place our electrodes and we look at the waves, they primarily are propagating in that direction. Another patient with gastroparesis, which just means delayed gastric emptying, the direction towards the small intestines is 180 degrees, but almost none of the waves are propagating in that direction. So that means that rather than the boom, 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 there's all this chaotic sort of spatial propagation, and that might be why you have nausea, possibly reflux, and it's taking longer for your stomach to empty. So this didn't occur in 2 patients. We actually took a look at patients with both gastroparesis as well as something called functional dyspepsia, which means you have all these symptoms, but you don't have delayed gastric emptying. And at the time, as we all know, the purpose of a doctor is to make you feel better. That's the job of a doctor, and yet previously all of the objective, non-invasive measures of gastric function did not correlate with the severity of symptoms. So here we got a clinically indicated set of symptom questionnaires, and our measure of the percent of time that you had abnormal slow waves correlated with the severity of symptoms across many categories. Stomach fullness, excessive fullness, stomach visibly larger, abdominal pain, abdominal discomfort, etc. And so if we summarize all that in just one plot, the horizontal axis is the percentage of abnormal slow waves, and the vertical axis is a summary between 0 to 5 of all of those questionnaires, 0 being small, 5 being more severe. So this was really exciting, and we've done more work in that area to further tease out the etiology of symptoms, but for the purpose of time, I won't go into that. Now, another aspect is that we originally were doing these studies in our GI collaborators' office using a $30,000 EEG amplifier. Could we actually miniaturize this, because a lot of symptoms can be transient. So imagine you waited for 6 months to go to this, GI specialist, and when you show up that day, your symptoms are not present. That would suck. So, in the spirit of, this analogy was more appropriate when I was at UC San Diego, where we moved a couple of years ago, riding our surfboard over the wave of other people's wave of innovation. We know when the Brain Initiative occurred, there was all this interest in miniaturization of technologies. You just heard the previous speaker. So we wanted to ride our surfboard over that wave of innovation, and there were all these miniaturized EEG recording devices that had been developed. So we basically hijacked some of these devices and MacGyvered them to see if we can get them to work for recording very low frequency signals of the stomach, and we were successful in doing so. And so we paired them with things like a very simple event logging application on your smartphone to dictate when you have a meal, when you have symptoms, when you have a bowel movement, et cetera. Implemented some of our artifact rejection algorithms that I talked about before, and lots of fun things occurred. We can consistently pick up that 0.05 hertz signal. We can see how the amplitude changes and fluctuates up and down over time. And if, for instance, as I mentioned before, a form of stimulus to the GI system is a meal, we can do things like looking at a meal response, and we can track how the stomach sort of increases the amplitude to process the meal and then gets back to baseline afterwards. And we can actually also pick up things like spatial information, as I alluded to before, which correlates with symptom severity. So one of the other interesting findings, if we begin to think about the connection with the brain and whatnot, was we got interested in looking at so we know that the stomach receives these parasympathetic and sympathetic inputs, but that's not the only organ that does that. So can we get a measure of, are things going wrong with you because your stomach's not operating correctly, or maybe the inputs to the system like sympathetic and parasympathetic are off? So we tried to take advantage of the fact that when we place electrodes on the abdomen, the biggest signal we actually get is the heartbeat. We have to filter it out. Can we take advantage of that information? So we looked at sleep versus wake, because we know when you're asleep, that's a setting where there's a lot more parasympathetic innervation, and we want to get a sense of the regulation of the autonomic nervous system. And so we developed some heartbeat dynamic measures, and we looked at how they occurred during sleep versus awake. And many people here are familiar with the LF-HF ratio. The numerator is sympathetic, the denominator is parasympathetic. So if there's more parasympathetic during sleep, what you should see is this number beginning to go down during sleep. So we were able to do that in a healthy human subject. You see it significantly does. We also had patients with diabetic gastroparesis. You saw virtually no change. So what we tried to do was to combine the stomach information. In particular there, we looked at the meal response, pre and post meal, how much of there was an increase in amplitude and a measure of its regulation, so intensity and regulation of the stomach from meals, but then also autonomic information, looking at sleep versus wake, intensity and regulation. And when we take those 4 features and we merge them together, using a very simple classification scheme, you can do things like separate healthy patients versus controls in 17 of 19 patients. And interestingly, as some of us know, about 50% of type 2 diabetics go on to have gastroparesis. So it's easy to determine if someone has diabetes. Could you, imagine the future prospectively being able to determine or predict the likelihood that maybe they're going to go on to have gastroparesis? So it turns out 9 of 10 diabetic patients we could separate in terms of gastroparesis or non-gastroparesis by looking at both of these measures. So, just a small little fun fact. We went beyond just MacGyvering an existing system for portable monitoring using off-the-shelf 3M electrodes, and we wanted to envision having just a simple patch like a Band-Aid that you place on the abdomen to do that using some of those flexible electronics technologies. And I actually was interested in doing this since 2011, but it turns out the approaches that we used with the John Rogers team to measure the EEG in the brain cannot directly apply to the stomach, and it's because of the fact that this is a very, very low frequency signal. And so gold type of electrodes are not going to work at very low frequencies. So we had to develop some clever ways to screen print silver chloride, but still get the benefits of stretchability. We finally sort of figured it out in 2021. And something else that we leverage is, rather than depending upon a CT scan, riding our surfboard over other people's waves of innovation, there are now little portable ultrasound devices you can plug into an iPhone, and it turns out that you can locate the antrum of the stomach with those. So combining those two things together, imagine just slapping a patch on the abdomen. So we moved to Stanford in 2021. We're fortunate to be in this new neuroscience building, and in the spirit of being in a new place, you kind of want to explore doing new science. And so one of the things I sort of reflected upon was, well, I started my neuroscience career studying the brain. And then I did this deep dive into gastroenterology in the gut, but increasingly we see these two things are connected. Now, a lot of people have tried to study the connection between the gut and the brain with things like the microbiome and whatnot, but I'm sort of this electrical engineer / neuroscientist. And given that we had this electrical perspective on the stomach, can we look at their interconnection? So we got a little bit about the interconnection electrically between the stomach and brain is that we have our enteric nervous system, we have our stomach, and as I mentioned, the stomach is the most vaguely innervated GI organ. And so afferents going from the stomach to the brain primarily first go through the brainstem and the nucleus tractus solitarius, and then go to some deeper brain areas, and the first cortical area that's hit is the insular cortex. And so this is an image from a nice review paper from Terry Powley, who I'm sure many people in the audience know. Now, in terms of downstream information, so that was sensory, motor information goes through the nucleus tractus solitarius as well as the dorsal motor nucleus of the vagus. And in fact, there was a very nice PNAS paper that just appeared in 2020 with viral tracing to look at all of the cortical areas that send downstream parasympathetic control to the stomach. And indeed, the anterior insula was found to be the dominant area to do that. So this is a little bit about the interconnections. And so, interestingly, complete coincidence, as we started to play around in this area, we discovered this group out of France, by the group of Catherine Talon-Baudry, discovered that there's an electrical coupling between the stomach and the brain that you can measure non-invasively. So they did a study where, using the same electrical measure that we do of the stomach and MEG to measure brain signals, what they found was the phase of that very slow rhythm of the stomach actually modulates the amplitude of the alpha band of brain rhythms. And so that's the good news. The bad news is that MEG, if we focus just in America, in 38 states in America, there's no clinical MEG. So relying upon MEG to understand these couplings is going to be problematic. So it's not surprising what we tried to do. We tried to explore doing this with EEG. So we have a paper that just appeared in December where we replaced MEG with EEG. And basically where you see the phase amplitude coupling in the alpha band in brain region specific areas, at a very high level we can get similar types of information, one being the right insula and the other being sort of parietal occipital areas. And we have ongoing studies doing this now. So this topic is the topic of this conference is neuromodulation. I've told you a lot about recording. What types of things can we do with neuromodulation? So one of the things that we got interested in doing was riding our surfboard over other people's waves of innovation, and there's been all this innovation in developing pacing devices for the heart. Can we repurpose some of these devices for pacing the stomach? And so we were able to do this in a bioelectronic medicine paper in 2021, where we basically developed an inductive coupling approach where you can power basically non-invasively, and you just have a very small device that can be inductively coupled. You can actually place this into a vein, and you can, we showed that in a study in pigs that we can actually pace the stomach. So we can deliver these pulses and pace the stomach, and all of the, the pulsing and whatnot that we did was within clinically acceptable sort of limits. Now, another thing that we tried to do, and our vision was that since you're inductively coupling non-invasively, Could you imagine that you just have this very small pacing device right there, but you can measure the readout about the efficacy of the pacing just with cutaneous electrodes? So in order to study that carefully, we measured right on the stomach, we measured the direction of propagation on the stomach, and then simultaneously cutaneously. And at a very high level, what we find is of these two plots, the top one is cutaneous, which is our long-term vision, but we also have the serosal recordings, which are right on the stomach. And where the red vertical line is where the pacing began. And if you focus on the bottom plot, what you can see is the wave directionality. It starts off being a bit all over the place, but what you can see is it starts to entrain a specific direction of the slow wave, as shown with the spatial histogram. So remember, this is on the axis of the stomach, and if we look back non-invasively, cutaneously, we see a similar effect where we see that the wave direction has begun to normalize, and I also communicate that more clearly in showing that the variance in the direction has significantly been reduced. Now, these two spatial histograms are not exactly the same directionally, but remember they're on two different axes, or they're on two different planes, three-dimensionally. The key thing is that they're narrow, they have low entropy, and they're consistent. So this is quite exciting, and we also got interested with some collaborators at UCSD, and I'm deliberately giving a shout out in the bottom right to Troy, Yifeng Bu, who I think is in the audience. He's presenting a poster, so I want to give a shout out to him. There he is. So this was worked jointly with some of our UCSD friends. And Troy's an electrical engineering PhD student at UCSD, and he has a couple of posters. And one of the things we wanted to do was to see, can we actually try to pace the stomach non-invasively? Can we give electrical stimulation cutaneously? And again, we did some imaging with this butterfly device to make sure we're targeting where we're delivering the stimulation. So we localized the fundus, developed stimulation to the lower sternum, and basically with short pulse intermittent stimulation, we can basically modulate the amplitude of the gastric slow wave, and in particular significantly reduce it. Something else that we got interested in is we know that we have this highway in between the stomach and the brain, and I mentioned, for instance, the vagus and the brainstem areas that it covers. We got interested in a variety of ways that we can actually measure this information non-invasively. Some people in the audience may be aware of optically pumped magnetometers. These are novel technologies to try to record magnetic fields. It's almost like the equivalent of magnetoencephalography, but it can be much smaller and it can be portable. You don't have to worry about spending so much money for one of these. It can subtract out the Earth's magnetic fields. And so we basically did a controlled experiment on the peripheral nerve where we stimulate, and we first just look at the effect of the action potentials propagating using standard electrodes, and then showcase that we can get the same information with optically pumped magnetometers as a first step in a controlled study. Something else we did, going back to the idea of these stretchable electronics, in another publication, we showed that we can pick up basically cervical compound action potentials propagating up the nodose ganglion. And in order to do this, we basically use one of these stretchable electrode arrays, carefully placed, and we basically interrogated the autonomic nervous system in multiple ways, with the cold pressor test as well as with a timed respiratory challenge. And by looking at these sort of multi-unit activities and doing some custom spike sorting, we could basically classify different firing patterns and biotype-specific changes in the nerve activities. So this was recently reported in scientific reports. And so, just to summarize, my path is really unique. I'm a bit of an outsider speaking to you guys. I'm very eager to look at all the posters and to get the perspectives of people who've been working on this their whole careers. But I started off as a quant, then I became a brain guy, then I became a digestive system guy, and now we're sort of looking at the coupling between them and the information highway in between them through the peripheral nerves, and looking at clever ways to not only record but also to stimulate, to modulate function. And be easy on me, because remember, I'm a newcomer to this field, but I'm really excited to be here, and I want to thank Hubert for inviting me. And a fun fact about Hubert, so he did his undergrad at UC San Diego, where I was for 10 years, and he did his PhD at Michigan, where I did my undergrad. So go blue. Happy to be back in Big Ten country.
So for that fantastic talk. I think we have time for a question or maybe two.
And all the tough questions we send to Troy.
Yeah. Okay, I would ask a question actually. If you can talk to us a little bit about the challenges that you have with those flexible systems. This is quite different from the first talk that we heard, very invasive, and now some really exciting work that we're getting with these non-invasive electrodes and especially those flexible systems. Can you comment on any of the challenges that you have with those, or where you need some new innovation there?
Yeah, so as I mentioned, it took us about a decade to figure out how to record the gastric signals non-invasively, because the fabrication procedures that we use, sort of the John Rogers style methodology, the types of metals that you can deposit in the clean room are quite limited. So for these very low frequency signals, we want to have more ionic types of measurements. So trying to figure out how we could do that non-invasively took us a while. And we kind of took advantage of the innovation of others, these new screen printing approaches. So we first fabricated with gold and chrome types of materials in the clean room, and then afterwards we're able to screen print the silver chloride, so with interfaces with the body does that. That was one type of challenge that we eventually figured out. One of the challenges that we have moving forward is that we now have so many people who want to partner with us to do these studies, and we're limited by fabricating these things in the clean room. So one of the things that we're spending a lot of time doing now are thinking about how can we leverage, and this is kind of analogous to the previous speaker's talk, there's so much innovation with these thin printed circuit boards that outside people in industry can do. To what extent can we leverage that so that we can only use our special procedures and as little as possible, and really leverage the scale of outside industry? And we have some unique approaches that we're exploring. In fact, we just sent something out for fabrication just earlier this week. So that's one area that we're really interested in doing, is trying to develop these very thin, flexible, adhesive integrated systems in a way that's compatible with large scale outside manufacturing, and there appear to be some opportunities. Now, another challenge with the work that I showed on the neck and trying to get this nodose ganglion type of information is everyone in the audience knows there's so many branches along the vagus. How can we be specific about what we're monitoring? And so I think that's going to be a combination of thinking carefully about the anatomy, computational modeling, simulating how the electric fields occur, and then how can we allow that to optimize the placement and the geometry of the individual electrodes. That's also an existing challenge. So yeah, I hope that was a helpful
excellent.
response.
Thank you for that.
I believe Hubert may have had a question.
Do we have time?
Keep it quick, Hubert.
Because you're next.
Okay, I'll cut my talk short.
Yeah, cut into your own time. That's fine.
No, Todd, I love your work, and I've been following it for many years. We're trying to translate this ultimately to people and to patients, and the whole thing I love about non-invasive is you're going to scale it out, right? I mean, this is the thing. Can you say a little bit about how sensitive it is to where you place these electrodes? Can you get it to like a smaller area, and could you use like electrodes, like references in different parts? Maybe it's on on a watch, and then you've got your patch that's going to be in a certain place, and can be easier. Is there still quite a bit of complex process, because it is distant, and I think it's impressive that you could do that, but how can you move it to that side, if you have any comments on that?
Yeah, so if you remember early on, I talked about the approach that people used in the 1980s. Part of the reason it didn't work is because of the variability with the stomach. So what we've been able to do right now is to develop an approach where maybe you can use a portable ultrasound to locate and then place your patch. If you looked at our patch wasn't that big. It was about the size of this. Now, another approach you could take is to use a bigger patch over the abdomen, and then basically with signal processing figure it out, because the further your electrode away is from the stomach, the smaller the magnitude of the signal you care about. There's pros and cons to that. Do you want to have a big patch over all of your abdomen? So those types of things we're still trying to figure out. But one fundamental issue is that there is a lot of variability of the stomach relative to landmarks that you look at, like the umbilicus or the xiphoid. Is that helpful? Yeah. Definitely. Yeah.
Great.
Yeah.
Thank you so much. I wish we could pick your brain for quite a lot longer. Thank you so much, Professor Coleman.
Thank you.
Really appreciate that. Thank you. Okay, and our final speaker of this session, I told you it was going to be a good one. Our final speaker of the session is Professor Hubert Lim here from the University of Minnesota. He's the Endowed Lyons Professor in Otolaryngology in the Department of Biomedical Engineering. He was hired as an Institute for Translational Neuroscience Scholar. He currently holds the Endowed Lyons Professorship in Otolaryngology. He's co-director of the Center for Neural Engineering, completed his BSc in Bioengineering at UC San Diego, followed by a dual master's in BME and EE and computer science, and then PhD in biomedical engineering at Michigan. His lab's research focuses here on neural engineering, neuromodulation technologies, sensory neuroscience, neuroplasticity, and neuroimmune physiology, with the aim of developing new electrical and ultrasound stimulation treatments for hearing disorders, pain, and inflammatory conditions in collaboration with multiple clinicians and companies. He's involved in 2 startup companies, serving as the Chief Scientific Officer of Neuromod Devices, developing tinnitus treatment device, and the Chief Scientific Officer of Second Wave Systems, developing wearable phased array ultrasound for treating various immune and pain conditions. Very much looking forward to your talk, Hubert. Thank you very much.
Thank you, Sarah. Normally I would start the talk saying I want to thank the organizers, but that'd be weird because I'd be thanking myself. So I'm going to actually thank the previous speakers, wonderful talks, and all of you for making it this morning. It's always tough after an intense day, previous day, and then staying up late and chatting and connecting with people. So I want to share today, it's a journey. It's been kind of an interesting journey and brought me, I think, Todd, to your point, feeling comfortable, feeling not uncomfortable, right? It's been a journey about 7-8 years, trying to figure out ultrasound and using ultrasound to modulate or activate the brain, the nerves, and organs. There's a lot of excitement around it, but a lot of also confounding factors and confusion. There's potential, but things that we have to work out. So I wanted to share that journey a bit and to also kind of give you some insight on where that field is, especially for the autonomic and organ modulation realm. Disclaimers, I have to show this. The Second Wave company will have some conflict-related things that I'll present. Tinnitus company, it won't be today. And then this other one, it doesn't matter because I don't have any money in that company, so it's pretty much irrelevant. And then some funding agencies here. So why would people even care for ultrasound, right? I mean, we know it for fetal imaging, we know for diagnostics, and where this all comes about is, we have these amazing technologies. You saw some presented, by Dr. Solzbacher, in terms of interfacing with the brain. And, it is invasive. It doesn't mean it's not possible to scale it up, because we know in the field I work in, cochlear implants, it can be very well done. But still, it is invasive, though you can achieve targeted activation. On the other realm, you have, on the extreme, is non-invasive technologies, and you're already seeing things that, Dr. Todd Coleman had presented. You can use magnetic fields, you can use electrical stimulation, TENS units, right? And so it is non-invasive, but there's always been a challenge of the specificity, and there's some things that, you can do to get around that, but there are some inherent, physics limits, right? So ultrasound came along. It's not new. It's, people have been doing this for 50, 60 years, and then, even in non-neural space. But in the neural space, it really started to pick up about a decade ago. A lot of people were getting into this. And the idea is that you could use beamforming, kind of like surround sound, speaker systems. When you're watching a movie, you can beamform the sounds. But you could do the same with ultrasound, and ideally beamform, through the skull, target different brain regions. But you could also do the same thing in the body, right? So there was a lot of excitement, and the question then became, through all these publications coming out, does it really work? Seems too good to be true, and things like that, try to figure out, are there confounding factors? And, I don't want to spoil the take-home message, but, basically, it can work. Some effects can be subtle, but other things that can be quite potentially effective and impactful, but it's just understanding what it's doing, right? And it's not just neural cells. There's also non-neural cells that you can modulate. I just want to showcase, it's hard to keep up because there's like tens of papers coming out all the time very quickly. But I just want to show there's lots of animal data, different types of studies. Most have not been invasive recordings and doing ultrasound and looking at the actual spiking pattern in vivo models. And that's what I'll show a bit today. But also because it's non-invasive, it's ultrasound. People are comfortable with it. It's been, lots of human studies going on now across the world, right? And especially outside of US, where there's a lot more accessibility and openness to using this ultrasound technology. So this is the paper that my colleagues and I published. It really kudos to Hung-Soon Guo and Mark Hamilton and a few others in the lab. I was interested in using ultrasound to treat tinnitus. It's ringing in the ears, and my target was the auditory cortex. And that was the idea. We spent 2 years. We had some challenges, and I'm going to share that. We found out heavily that there was an auditory effect created, and I'll show you how that happens. Separate group, Dr. Mikhail Shapiro's lab at Caltech and Doris Chow, they were running into similar things, so we decided let's publish together so that people could better understand, what's going on here. So what we did in that experiment is that I was trying to stimulate the auditory cortex in guinea pigs, and the idea was that I was going to record also the neural activity in the auditory cortex. So I want to see how we're able to modulate the activity and suppress even activity. We can create some hyperactivity to mimic tinnitus. Don't worry too much the other part, but I was going to map out the connections from the auditory cortex to the auditory midbrain downstream, because we know that there's connections in that pathway. So it was just a way for me to kind of show that I could move the beam and shift the patterns of activity downstream. Now what we did was we applied ultrasound to the brain, but we also could do sound stimulation to the brain. And as you see here on the left side, I don't want to be too complicated here, but it's a multi-site electrode array. So we've got 4 shanks, 8 sites along each shank, so 32 groups of plots here. One is the local field potentials that you can pick up on the site, and the other one is a histogram of the spiking activity that you get if you do 100 repetitions of a given stimulus. So the main thing to take home here is that we've got these 32 pairs of plots. You do sound stimulation, you see what's on the left. You do ultrasound stimulation of the brain. It looks very similar, so we're like, oh, that's kind of weird, right? Even the latencies were similar, and you would expect that the latencies would be shorter because you're stimulating the brain. So the obvious thing is, okay, the animal's hearing this, right? So what we did was we decoupled the ultrasound transducer from the animal, and the activity went away. So it wasn't being produced by the ultrasound transducer. It had to be on the head. Well, then we said, okay, well, let's just try to put the ultrasound in different places. So we put it on the auditory cortex, record from the auditory cortex. We put it on the visual cortex, record from the auditory cortex. We put it on the somatosensory cortex. We put it on the skull. We even put it on the eyeball, right? I'll get to why we did that. But we put it on the eyeball and even on the body. And the point here is that, recording from different cortical layers, you don't have to worry about the complexity there. But the point is, in animal 1, you can see in all these different positions, everything kind of looks similar, right? And then for another animal, things look similar. Well, this made me realize, because when I was doing my postdoc in Germany, I was in one of the biggest hearing clinics. They were working on bone-anchored hearing aids, and they had these head screws that go in, even in pediatrics, that are drilled into the skull for a hearing aid. And sometimes they get infected, so they drilled that out, and they did some experiments where they just vibrate the dura. And you can actually hear when you vibrate the dura. And you can also, I wouldn't recommend this, you could put a vibration device on your eyeball and you can listen to speech, right? So the reason why that happens, though, is because the fluids in your brain, the CSF, is connected with the cochlear fluids through what's called the cochlear aqueduct. So it's all continuous. So you can actually hear through vibrating the cerebrospinal fluids. Anyhow, this made me kind of think, oh my goodness, are we just vibrating cerebrospinal fluids? So what we did, experiments, we did many controls. We basically cut the auditory nerves and found out that when you stimulate with ultrasound, all that activity disappeared, right? Now, we tried for 2 years. I have to say my PhD student, Hung-Soon Guo, he's at Caltech now. His whole PhD was, I tried everything, but I got nothing. And that was basically his PhD, which was great because he got a neuron paper out of it, a scientific reports, and he got a postdoc at Caltech. Not really the ideal situation you want to have for your your PhD thesis. But it did show us that there are a lot of parameters, and the question is, is there still effects going on? I'm not going to say that it's not working. It's just at least obvious excitation effects were not possible. And just to give you a visual here, the way we believe ultrasound is causing a lot of activity, not the only activity, but we found out that at least in rodents, it's like a jet engine loudness. So you're basically doing ultrasound through the cerebrospinal fluid to the cochlear fluids, and then it causes equivalent to like over 100 dBSPL. You're basically activating the entire brain, so you're going to get activity wherever you record from because of cross-modal projections. So this is what we believe is one pathway that was happening. And I don't want to go into this too much detail. We did lots of controls. We even took a dead animal and a live animal, connected them through a fluid pathway, and showed that we could get activity in the live animal by stimulating the dead animal through a fluid pathway. And these are just different experiments. If you're interested, they're all in the paper. So where does this put us then for ultrasound? I think the community is realizing that there are a lot of confounding factors. And if there are modulatory effects, they're not going to be huge effects. And even this paper came out, they said, however, the enthusiasm about the neuromodulation, modulatory potential of ultrasound has recently been dampened by studies. They're talking about our studies. We dampened the field. But in addition, such overt effects have not been observed in large mammals, including humans, only small changes in neural signals and so forth. But they did do a seminal paper here where they did show that they could make behavioral changes, right? And through additional studies, what they're finding now is that there is a depolarization effect. And I'll show you another paper after this slide. It is causing depolarization effects. They're not readily leading to action potentials, and it seems to be one pathway is a calcium channel activation, but it's been hard to get spike potentials in vitro, but also in vivo. And I recommend you read this paper. Our colleague Mikhail Shapiro, he's showing in one mechanism, not the only, that ultrasound can activate calcium channels. Those lead to opening of other calcium and sodium, voltage gated channels, and eventually the idea is that it could lead to action potentials, right? But I want to kind of normalize this a bit here, because we got, it's kind of like you get excited and you're like, I'm the smartest person in the world, I'm innovative, and then you realize someone did it like 30 years ago or 40 years ago, and you're just like, wah, wah, this was not so. But if you look back at literature, I mean, there's already been like 40 years of people doing ultrasound, and not only neural cells, but in non-neural cells. And there's a whole realm of opportunities there because they're modulating endothelial cells, stem cells, chondrocytes, sensory receptors, right? In addition to neural cells, and there's a cascade of, signaling pathways that happen. So that's, the exciting part of cells. Now, that was the brain side. I just want to highlight on the nerve side, we also did the same thing. And Hung-Soon Guo, another paper where I tried everything, and I found, well, he did find that inhibition is possible, right? So I do want to give that nugget there that's important. So what we did in these experiments is that there were papers coming out showing that you could use ultrasound, but they were focusing on non-invasive stimulation and recording non-invasively EMG, right? And so we know that ultrasound can activate skin receptors, muscle. So we wanted to make sure that what they're activating was truly the nerve. So we did try to recreate what they did, ultrasound of the leg, in this case, sciatic nerve. And we have another 32 lecture in the somatosensory cortex. We're able to record, and these are just post-stimulus time histograms, spiking activity, and you just see that there's activity. It's biased to one shank. It's 4 shanks because there's a topographic map of your body in the somatosensory cortex. So you would see if you're stimulating the leg that the activity is more in the leg region of the somatosensory cortex. We remove the skin muscle above it, just trying to get onto the nerve itself. And we got stronger activity. So we're like, oh, wow, this is working to activate the nerve. But then when we removed it and coupled it, the nerve by itself, isolated with the parafilm film, we found out that the activity was gone. Now, being critical of our results, well, what if we just damage the nerve? The nerve is not functioning anymore. So, of course, what we did then is we stimulated that nerve. We recorded from the other side to make sure the nerve was functional, did the ultrasound to make sure whether or not it's activating, and because there's artifact issues too, we stimulated the foot and recorded the nerve activity. So we did these kind of different controls. And what we saw is that nerve stimulation with electrical current, it's a little bit hard here, there's an artifact, and then you get the evoked potential average, the single trial is the red. You get post-stimulus time histogram in the somatosensory cortex. You stimulate the foot, you get an activity a little bit delayed, obviously, because it takes longer to get to the nerve from the toe or the foot. Activity, we did ultrasound, nothing. We tried over 100 parameters, and only when you blast a ton of energy into the system, you can activate it, but by then the nerve in the leg's moving, right, with ultrasound. And so I don't know if that's just vibration of the leg or it's ultrasound. So these are some things that we discovered, but on the other side, what came out of it was we did find that you could actually inhibit nerves. And the important there is that, with electrical stim, it's always challenging to be able to shut down nerves. You gotta use like high frequency stimulation, put it into refractory state, but with ultrasound, there's a potential here to use it as a complementary tool to electrical. So in this case, because these experiments, we're trying to keep it simple, we stimulated the foot, recorded the nerve activity on the other side, we did ultrasound, we did the foot stimulation to cause the activity, and then while the activity is ongoing, we did the ultrasound to see if we could suppress that activity, okay? So it's happening simultaneously. And you can see the black curve here is the non-ultrasound situation, evoked activity, and then the red, you could actually suppress it down. And what we did, x-axis, you could think of it as some kind of amount of energy we're applying, right? The y-axis is lower means that we're suppressing, going negative, and you could see that for all many different parameters, as you increase energy, the activity decreases, but also the thermal effect, it's a temperature effect, is also increasing, right? So there's gonna be a sweet zone of where you can achieve that. And it's not only the nerve, the brain also similar. We stimulate, any brain region. We've tried many, and it'll be published soon or submitted soon. We recorded the activity, in this case, somatosensory cortex to foot stimulation, and we got activity. Because of confounding auditory effects, we've done controls and so forth, but it was just a pain to do it, all these controls all the time. So we actually just stimulated ultrasound in the middle, and then we did the recordings before and after, so there weren't kind of these confounding factors simultaneously. But you could see there that the residual effect, you do get a suppression of the activity, the red curve, and if you go to higher duty cycles, more energy, and again, this is a thermal effect, you're able to suppress it. And you could actually suppress it down to 0, but I will tell you, many of those times, those zeros never came back. So there is some question of safety in terms of how much energy you can drive in, how much you can suppress, and then how much you get recovery. And here it's just showing that there is recovery, but there's some residual effects that are happening, as you could see here, where we have, the post-ultrasound drops down, and it comes back a little bit after a few minutes. I just want to give a shout out to Tay and David Darrow. You saw a talk. They did a paper in 2019 also showing that you could do ultrasound. They did non-invasive prep with EEG activity as well, and they were able to show with focus stimulation also appearing to be a thermal mechanism. So that's great for the brain and nerves. There's opportunities there. You could use it for scientific experiments. You're not going to get very obvious excitatory effects, but at least for modulatory effects and inhibitory, those are some options there, right? So let me shift gears now for the body stimulation and organs, because this is where I think it's going to get very, very exciting, right, in the upcoming years. The question, and it's because ultrasound is being shown not only for neural cells, but also for non-neural cells, right? And so the question that my group was interested in, and this was funded through DARPA, and collaboration with Medtronic and eventually with Second Wave and Ultran Group, we want to see can low-intensity ultrasound, so when we say low intensity, we're still talking about within the safety ranges of diagnostic ultrasound imaging. Can we stimulate, in this case, the spleen? And you saw a great talk by Dr. Sangeeta Chavan in terms of the immune-brain axis, but can you stimulate using ultrasound to modulate the spleen to control immune? And I don't need to go through this slide because she did an excellent job yesterday, but we know that there's this interaction between the brain and the spleen pathway that can help regulate, or in what we're interested in, dampen the inflammation in the body. And one pathway there is the vagus nerve to the spleen. And as Dr. Sangeeta showed, basically that you could use this pathway to be able to shut down the hyperinflammation, for example, in rheumatoid arthritis patients, and there's also results that are coming out for other conditions as well. So what we did is we stepped back a little bit because no one had done ultrasound of the spleen, at least we didn't know of anybody, to treat inflammation, and this was a DARPA-funded project. And actually Dan Zaks is here in the audience somewhere, and a few others, Claire, Rachel, who's not at this meeting, but many people and Medtronic folks as well. We approached this project and we're just trying to see, is this even possible? And we were fortunate to get positive results. The thing is, ultrasound of the spleen treating inflammatory arthritis sounds a bit out there. Fortunately, our program manager, Eric Van Giesen, connected us with GE folks because they were working on a similar project with Feinstein Group, and they had found similar results, but using a different animal model, a sepsis animal model in rats, and we were using mice arthritic model. So what we did in our study is we basically took this model that my colleague, Dr. Bryce Binstad, had developed. It's just a great model. It's an arthritic mice model where you can basically inject the animals serum from healthy animals with serum from animals that already have arthritis, and then they develop arthritis over time within 7 to 10 days. And so we can then take those animals, do ultrasound stimulation directed towards the spleen, and then try to access this anti-inflammatory, cholinergic anti-inflammatory pathway to dampen the immune response. This is just showing you that model where it develops over 7 to 10 days. And then what we do is we take this ultrasound transducer, there's a cone, which just helps us to figure out where the focus, the highest energy point of that beam is, and it's targeted over the spleen. And then we can basically stimulate over days, right, relative to them being injected with the serum that causes the arthritis to develop. And then, of course, we can take blood samples at the end, and throughout that process, measure the effects of ultrasound on the swelling and arthritis. So there's two measures we use, ankle thickness. Obviously, you want the ankle thickness to be less, You don't want it to keep getting thicker. And clinical scores, you can look at the joints in the hands and the paws to basically see how much swelling or just abnormal configuration there is due to the arthritis. And using a parameter that we found to be one of the more effective ones, you can see here that with the ultrasound, we're able to improve the ankle swelling as well as the clinical scores. And this was the one that was most important to us, is that there was a dose response effect. So the more ultrasound you provided, from 2 minutes up to 20 minutes, and people keep asking, well, what happens after 20 minutes? And I don't see Dan. Well, maybe Dan's in the audience. Well, Dan's there. He would have basically have to have worked 40 hours a day because he had to take all the animals and then do the ultrasound in a sequence pattern. So this was actually the maximum logistically we could do. But there's no reason to say that more energy might not lead to more improvement, right? So that was exciting for what we had done in a more chronic-like animal model. What G had done and Feinstein group, they were able to, it was a beautiful paper, you should check it out. It was a companion paper with ours. They did a lot of control experiments and mechanistic studies. And I don't have time to go through it, I recommend. But what they found was, it's not the only pathway, because not all the effects proved to be just here, but it was a big portion of it. It ultimately led to this interface here, which is when you do ultrasound, and just to reiterate what Dr. Siobhan showed yesterday, is you have the splenic nerves coming into the spleen. They're going to release norepinephrine to activate T cells, and then the T cells will release acetylcholine to then dampen the macrophages, right, or the immune cells. And so where the ultrasound appears one spot of causing this effect is, as I told you, nerve activation is not so easy. So my belief is that it's neurotransmitter release, because I showed you that calcium can be, the influx can happen due to ultrasound, so there could be neurotransmitter release. But also, it could be that as there's ability to modulate non-neural cells, there's also the ability to do ultrasound in these different cell types, right? So does this work in humans, right? That's the next big question. All this animal data, things are building up. And so, fortunately, Sangeeta presented yesterday ultrasound in humans, at least in healthy humans. That has been shown to decrease TNF, so an inflammatory marker. And this, before we were trying to do a paper together combining some of our human results and theirs, Nature reviewers didn't accept it, and then we decided to part ways and separate the publication. But then, as you saw, the paper was published just a week ago, right, Sangeeta? Yeah, so that if you're interested, showing in humans that you could do ultrasound of the spleen using the GE cart-based system to be able to reduce down the inflammatory marker TNF. We also ran during the height of the pandemic. There was some funding. DOD was interested to see if we could help dampen hospitalized COVID patients. And so we actually got ramped up very quickly. PPE, kudos to the people in my lab. They were able to get in there and apply ultrasound for up to 7 days over the spleen, and then treat patients who were hospitalized with COVID-19. And we did see some encouraging results that I presented before. I don't have time here, but we're putting it together in a publication that we should hopefully get submitted soon. And then right now, what we're doing is we have an ongoing rheumatoid arthritis study with ultrasound. This is the company Conflict of Interest Transparency, it's a company that I'm involved with. We developed a portable device that can be placed over the ribs that you can stimulate the spleen, and we're running an ongoing clinical trial now treating rheumatoid arthritis patients. And we're getting close to being finished with that, so hopefully I can present that at an upcoming meeting. So just the last few minutes here, just to keep us on time. Beyond rheumatoid arthritis, there are other applications that have shown to have potential. You can do ultrasound stimulation of the spleen to interact with the immune axis for treating acute kidney injury, right? So there's papers that are coming out related to that. And some of the seminal papers were actually started in that topic. Just recently, I just saw this paper. It's not published yet, but it's bioarchives. You could think about accessing that anti-inflammatory pathway for treating pulmonary hypertension with ultrasound, so that's exciting. The group, GE group, just have been so productive. They were doing ultrasound stimulation of the celiac plexus for a rat model of inflammatory bowel disease, and they're showing positive results there. And this is in animals, though, but still, it's not hard to imagine moving to humans. Now also targeting the liver, the pancreas, the GI tract. So many different opportunities here. And you might say, oh, does this actually work? Well, GE, I have to give credit to them. They were able to do a whole series of impressive animal experiments, small and large animal experiments, and then they've moved to clinical trials for doing ultrasound stimulation of the liver to treat diabetes, right? So this is already moving along very quickly, and the last thing here is you need technologies for it. And so fortunately, there are two major groups. GE is developing more portable systems, and our company as well is developing portable systems that can be applied all over the different body. In the future is the idea, and trying to provide a more low-cost, accessible approach to treating some of these things that would typically be treated with drugs. And finally, I know JoJo's here. If you're interested, we did a Scraps episode, myself, Chris, and Mikhail Shapiro. We talk about a lot of these topics, if you're interested. And then finally, that's the end of it, and I'll take questions. So thank you.
Good. Great, thank you so much for that great talk. Do we have any questions from the audience? Yes, go ahead.
Yes, hi, Hubert, very nice talk. I'm curious, have you, so Li Wei-Tai at MIT actually looked into like sound, some certain sound frequency that can induce lymphatic flow in the changes in the brain. I wonder if you have looked into sort of the lymphatics changes in the brain with your ultrasound stimulation, given your theory about might tap into, the spinal cord fluids and all that.
Yeah, so I do think there's possibilities that you can modulate those pathways, whether it's through a fluid pathway, but I even think just a cellular pathway, modulating cells that would be in those regions, right? And so because ultrasound is not only the mechanical effects that we see, because that's, I guess, unique to the auditory system, but all these other studies coming out are showing. And actually, Chris Puleo and GE, they stimulated directly the lymphatics also, the lymph nodes, and they're showing effects. So I think there's a lot of opportunities there. Yeah.
I think we have another question over here.
Hi, thanks, Hubert. This was great. What model or what kind of arthritis were you guys specifically studying? Was it rheumatoid?
So we don't call it rheumatoid. We call it inflammatory arthritis because of just the relationship. To rheumatoid arthritis is a bit, how do I say, loose. But yeah, we call it inflammatory arthritis. I would leave it at that.
Okay,
Yeah.
and then were you, you talked about ankle thickness basically, but obviously there's more symptoms and problems associated with arthritis, there's increased risk of osteoporosis and decreased cartilage, and did you guys look at any of those other markers?
We didn't in the animals. This is something that, this study we're running now is a pilot study, and that is going to look at some key clinical and blood biomarkers. But depending on how the study turns out, the plan is to run a pivotal clinical trial to get FDA approval for a device, and in that case, we are going to be looking at some of these things as additional endpoints or analysis endpoints, just to see what's going on for more informative reasons.
Very cool, thank you.
Great.
Thank you.
Last question right here in the middle.
Hubert, nice work. So we know that mechanical stimulation of a peripheral nerve can evoke ectopic action potentials, and it has to be very brisk. And what you're doing with ultrasound is very brisk, repetitive stimulation. Is there any way of getting a single pulse? So you just evoke, one ultrasound wave, which is very high frequency, high intensity. I would think that should cause direct activation of mechanosensitive ion channels, piezo channels.
Yeah, and they can actually, if you go high enough and you cause enough pressure inflection on it. Whether I would do that more than a few times would be the question. But I think scientifically, you could do this to run interesting science experiments, right? Where you just do it a few times to perturb a network, right? I think that's the beauty of this approach. And that's what I think where ultrasound could actually be used, at least in the brain, for a lot of interesting scientific questions. Because we would always love to deactivate a node of a network and see how much that's contributing to an effect. And I think that's where ultrasound could actually be quite powerful. Yeah.
Great. Thank you so much. Let's thank Hubert one more time, and thank you to all of our speakers in the session, and let's continue this in the break.